Okay. All right, so let's look at page 6B of your notes. It says protein synthesis part 3. Protein synthesis part 3. All right, so this is translation. Okay, and what is happening in translation? What are we doing?
We're making DNA.
Well,
Proteins.
we're making proteins from the DNA, and we're translating, okay? So we're translating from the codes that were on the messenger RNA to amino acid language, okay? So we drew a picture already of our ribosome that should look at least somewhat similar to this. You will notice here that this does require energy to take place, but instead of ATP, it's GTP. Both of those are energy-containing molecules, so it does the same type of thing, okay? It's triphosphate, it's guanine, it's made out of guanine, it's guanine triphosphate, whereas the other one is adenosine. So, anyway, you can see that it loses a phosphate to give off energy and becomes diphosphate. So very similar to how the ATP works. All right, so we've got our start codon. Your start codon is always going to be AUG. It's methionine, okay? Now, if you'll look here on the pink there represents the codon that's on the messenger RNA. The UAC that is attached to it represents the anticodon that is found on your transfer RNA, okay? And all the anticodon is for is, you see on your transfer RNA, you've got this amino acid hooked to the end of it. It is just like a key that fits to your codon to make sure that it's bringing the correct amino acid, all right? So it's just kind of a safety net to make sure that if those fit together, then you know that it has brought the correct amino acid. All right, so on your ribosome, you've got an E site, a P site, and an A site, and we mentioned that already. We've already talked about that. The E site is where it exits. The P site is where the protein is built. The A site is where the amino acids are added. That's where the new transfer RNA comes in. And it acts kind of like a conveyor belt. They're added at the A site, they'll move over to the P site where the protein gets made, and you'll just keep having amino acids added onto here because amino acid is a monomer for making a protein. And then when it drops off its amino acid, it goes to the E site and then exits. E stands for exit, okay? So that is initiation. That's how it gets started. Then you've got elongation. So what's it doing? It's making it longer, okay? All right, and that's it. What's happening is exactly what we just talked about. Initiation is where the process gets started with AUG, which is methionine, okay, that we just talked about there in the pink. All right, and so then after that, you notice on here that you've got these transfer RNAs coming in, see? And they'll attach, the anticodon attaches to the codon on the messenger RNA. A peptide bond forms between them, between these amino acids right here. It just drops off its amino acid, the one that was used there before. See how it just transfers over? You've got your amino acid chain there, and then it just transfers over. A peptide bond forms between them, and all a peptide bond is a bond between amino acids. Okay, that just means it's amino acids bonded together. So it is going to transfer that whole chain over here, and then this one, notice, doesn't have a chain anymore. Okay, this one right here. All right, and it's going to be shoved over to the exit site, and it's going to fall off because we don't need it anymore. We just needed it to bring in the amino acid that it was supposed to bring. All right, so, and it just creates then a chain. You've got this long chain of amino acids, which is what a protein is. All right, and then you get termination. Terminate means what?
To eliminate, destroy, don't get rid of.
Release. Okay. All right. So release, destroy, get rid of, stop. Okay. If you're terminated from a job, which I hope none of you ever are, but if you hear that someone else is terminated from a job, that means that they have stopped working at that place. Okay. So that is what happens with termination here in regards to translation. You actually have something called a release factor that comes in and occupies a space, but what does it not have attached to it? An amino acid. There's no amino acid attached to it. It's just a little shape there. And so it says, hey, we're done. Transfer RNA leaves. Here's your little amino acid chain or polypeptide, which is means many peptide bonds. That's many amino acids linked together, and that makes a protein. And then all this just breaks apart because it's done. Okay? All right, now you can have something called polyribosomes. What does poly mean?
Mini.
Many. So many ribosomes, and what they're showing you right here is that many ribosomes can actually be attached to the same messenger RNA molecule so that you can make lots of the same protein. If you need many of that kind of protein, they'll all just attach to the same messenger RNA and pump out lots of proteins all at the same time. Okay, so here's our big picture. We had transcription taking place in the nucleus. You've got your messenger RNA was made 5' to 3'. We had to do 3 things to it to modify it before it could leave the leave the nucleus. What did we put on the 5' end? A cap. Put a little cap on the 5' end. All these little light spaces here, the light pink, that's our introns. They're going to stay inside the nucleus because we're going to cut them off with spliceosomes. Okay, the exons are going to hook together. We're going to put a poly A tail on. Then that's going to leave the nucleus, go to the ribosome, connect to it. You got your transfer RNAs brought in through translation, and you're making amino acids. All right. Now, in order for the protein, do you remember that protein has to be folded in order for it to work right? Yes? Yes. Okay. All right. There's something called a chaperonin. Okay. What is a chaperone at a dance? What do they do? What's their job? Okay, they watch you. They make sure that you don't do anything that you're not supposed to do, and all that. Okay, so they're your monitor to make sure that you act right. There is a chaperonin inside your cell that it's a pretty cool looking thing. It looks like a capsule, and it will take your protein, this chain of amino acids, and these amino acids will go down inside of this and a cap's put on it. It folds up the protein like it's supposed to be to make it work, and then when the protein is folded right there, it'll leave. Okay, if this happens, and if you'll look on your notes, I think chaperonins are talked about on there. Yeah, down at the bottom. If you will box in the word in, if a protein goes into a chaperonin, okay, it is going to stay, that protein is being used inside the cell. So it's going to stay in the cell. It's never going to leave. All right? But if you'll remember when we talked about, and it mentions there the primary sequence, the one with the little thing at the top, that just means that it's not folded yet. It's just a chain of amino acids like that. All right, but if you'll remember when we talked about the cell parts, the rough endoplasmic reticulum is where proteins are transferred out of the cell. They're put in little vesicles to be transferred, or little folds through these membranes to be transferred out of the cell. So if the ribosome goes there to the endoplasmic reticulum to be modified, that means it's being prepped to go out of the cell. So it's going to transfer it out.